Good afternoon, and it's our privilege to welcome you to the Andel Institute Public Lecture Series, a focus on Alzheimer's, Parkinson's, and Lewy body dementia. My name is Brett Holloman. I am the Chief Development Officer here at the Andel Institute, and we are glad today to have this opportunity to share with you some of the research that's happening both inside of our facility and beyond as we reach out into the world. Today we have the privilege to hear from two of our most recent additions to the Institute. We will hear from Dr. Jose Bras and Dr. Rita Guerrero, two scientists who are working in the Institute's Center for Neurodegenerative Science to better understand the intersection between Parkinson's, Alzheimer's, and Lewy body dementia. They also work to discover new ways to go about diagnosing and treating these diseases. Dr. Bras is a leading molecular geneticist whose research focuses on genetic variability, impacts the onset and progression of neurodegenerative diseases such as Parkinson's and Alzheimer's. He earned his bachelor's in biomedical sciences from Coimbra Polytechnic Institute, followed by his master's degree in biomolecular methods from Aveiro University, and his PhD with honors from the Coimbra University as well. He conducted his doctoral research at the Laboratory of Neurogenetics at the National Institute of Aging of the National Institutes of Health, where he investigated the genetic basis of movement disorders. In 2010, Dr. Bras joined University College London as a postdoctoral research assistant in the Department of Molecular Neuroscience. And in 2015, he became a tenured principal investigator and was later named as a program leader at the UK Dementia Research Institute at the University College London. In 2018, Dr. Bras joined Andel Institute Research Center for Neurodegenerative Science as an associate professor, and it is our privilege to welcome him. Dr. Rita Guerrero is also a leader in parsing the genetic variations that contribute to neurodegenerative diseases such as Parkinson's and Alzheimer's. She earned her bachelor's degree in biomedical sciences from Coimbra Health Technology Institute, her master's degree in biomolecular methods from Aveiro University, and her PhD, also with honors, from Coimbra University as well. Her predoctoral work was conducted at the Laboratory of Neurogenetics at the National Institute of Aging of the National Institutes of Health. In 2010, Dr. Guerrero joined the Department of Molecular Neuroscience at University College London's Institute of Neurology as a postdoctoral research fellow, where she explored genetic variability in several neurological diseases, including Parkinson's and Alzheimer's, and also investigated how genetic variants affect the disease process at the molecular level. In 2015, she became a principal investigator in the Department of Molecular Neuroscience after holding a traveling research fellowship from Alzheimer's Research UK and obtaining a senior research fellowship from the Alzheimer's Society. In 2017, Dr. Guerrero was named program leader at the UK Dementia Research Institute at University College London. And in 2018, Dr. Guerrero joined Andel Research Institute's Center for Neurodegenerative Science as an associate professor. Today, we'll have the privilege to hear from both of these outstanding research faculty. We will first hear their presentation, and then we'll leave some time for questions and answers following. If you'd please join me in welcoming Dr. Bras and Dr. Guerrero.
Thank you very much. I really wanted to start by thanking the Institute, thanking Brett and his team, Kayla, Beth, Sarah, everyone that actually made this possible, made it possible for us to be here today, share a little bit about our work with you, and thank you for your interest in our work and for taking the time to be here today and hear about Alzheimer's, Parkinson's, and DLB. So, as Brett mentioned, we moved quite a lot in the last few years. We did our formal education in Portugal. We then did our PhD work at NIH in Bethesda, just outside Washington, D.C. We moved again across the ocean and we went to the UK and did our postdoc trainings at University College London. We stayed there for 8 years. We were named program leaders at the recently created Dementia Research Institute. And then we visited the Van Andel Institute, and we really enjoyed the environment here, how friendly everyone is here, and the collaborations that are going on here, and the great science. So we decided to move, and we moved late last year, and I made sure I would put this in writing. We have been here since December 2018, which means that we survived the winter here. And coming from the warm temperatures of Portugal, this is a big thing for us. And we were also very fortunate to bring with us three lab members. Celia, who is a senior research technician, she really makes our lab run smoothly, and she knows a lot about all the techniques that we use every day. Susanna, we've known Susanna for many, many years. She's a postdoc now. She works to dissect AD genetics risk. She focused on a particular gene that I'm going to talk a little bit about further in this presentation. And Nadia, who is a PhD student, and she also recognized the interest of this place and how wonderful this community is and how welcoming this community is. And she left a fully funded scholarship at University College London and started her PhD here at the graduate school at the Van Andel. We were also very lucky to have waiting for us Emily, who has been a lifesaver and has really made our lives much, much easier over the past few months. And we are really, really lucky to have all these people working with us. So what do we do every day? And in a very general and global way, this is how we see our work. So we try to identify genetic regions and genes that will then lead to a better understanding of these diseases, biological understanding of these diseases that will lead us to identify targets that can be used to develop treatments, to develop drugs that can cure, prevent, or delay the onset of these diseases. We focus mainly on this area here, and we rely a lot on collaborations to take our work further until treatments. More specifically, the types of studies that we do with different flavors, but basically are these two types, family studies and case control studies. What we do in family studies is to study the DNA, the genetic background that is present in family members that have a disease, and compare this DNA with family members that don't have the disease. And we try to pinpoint the mutation, the cause of disease in these families. And these are diseases that are passed through generations within families. We also do a lot of case control studies, and here what we do is to compare the genetic background of large groups of patients, of people that are living with a disease, Alzheimer's or Parkinson's, and we compare this DNA, this genetic background, with a large group of people that are matched in their ages that don't have the disease. And what we are trying to do there is to identify risk factors, so genetic variants that increase or sometimes decrease the risk for these diseases. And how do we do this? And I'll pass this on to Jose too.
So I'll just give you, I'll just show a few slides with some important aspects of things that we think will help in setting the stage for the rest of the presentation. So how do we do this work that Rita just mentioned? When we first started working in genetics, we were able to look at a single gene in a few dozen individuals. And really, if we wanted to look at any more people, this would automatically become, a month-long project, a multi-monthly long project, sometimes several years, to be able to look at more individuals. Today, we're able to look at all genes that exist in many thousands of individuals. And this is only possible because of the developments in technology that have happened over the last couple of decades. And they've really been transformative for this field. And so, of course, studying many thousands of individuals has the implication that the work that we do needs to be very collaborative in nature. We need to have access to all of this patient material, to all of this clinical information, and we can only do that if we collaborate with other researchers, and we have to do that at a global scale. And so here we're just showing some of our collaborations with whom, people with whom we work on a daily basis. And so really to show how collaborative our work is. So in setting the stage, we thought it would be important to just show a couple of slides of things that are more basic. And so what we do is study the DNA of human beings, people living with disease and people without disease. The DNA, as you'll know, contains the information to, in the end, make proteins, and proteins are the workers of the cells. These are what, proteins are what make things happen in the cell. And so when we have a change in the DNA, this is usually propagated into the protein. Now, these changes may have an effect in the protein, or they may not have an effect in the protein. This is an example of the beginning of a protein. Proteins are made up of amino acids. These are the building blocks of proteins. And the information for each one of these amino acids is in the DNA. And so we can easily understand if there's a change in the DNA, this will be propagated into the amino acid sequence of the protein. Sometimes this has implications for health, for example. So there are changes in the DNA that are rare, that we call mutations, and when they occur, they cause disease. These are a couple of examples of how these diseases can be inherited. They can be recessive, and if a disease is said to be autosomal recessive, this means that, as you know, every one of us receives 2 copies of our DNA, one from our father, one from our mother. In an autosomal recessive disease, for the disease to show, we need to inherit both copies of a faulty gene. So this is what characterizes a recessive disease. We need to inherit both copies, one from mom, one from dad, with a faulty gene. On the other hand, we also have dominant forms of disease, and in this case, a single copy is enough for disease to occur. These are called dominant diseases, and a single copy from either the mother or the father is enough to cause disease in the offspring. So these are rare changes in the DNA, things that are very rare that cause these types of diseases. There are, of course, also changes in the DNA that are much more common, and they tend to not cause disease. Otherwise, we'd all have all of these diseases. So common changes in the DNA tend to not cause disease, but they're very, very important. Sometimes they increase or decrease a person's risk to develop a disease. So they don't cause it, but they modulate the risk of a person to develop the disease. And this is an important aspect. But also important is the fact that not all DNA change is bad. In fact, we're all different because of mutations in the DNA, of changes in the DNA. If you think about it, we all share the same background of the human genome. We all have the same background of 3 billion letters in our DNA, but we're all very different from each other. And that happens because of mutation, because of changes in the DNA that make us different from each other. And so these were just a few slides that we thought were important in setting the stage for the things that we're going to talk about next. And I'll pass on to Rita to tell us a little bit of our history with Alzheimer's disease.
Okay, thank you. Okay, so talking a little bit about Alzheimer's disease, and I wanted to start by highlighting really the really heavy burden that this disease has in our society. We are talking about the 6th leading cause of death in the United States. In 2018, there were close to 6 million people living with Alzheimer's disease in this country. And these numbers are really expected to increase by a lot, and expected to reach 14 million by 2050. One other aspect that it's also very important, but it's many times difficult to account for, is how many hours of care is given by family members. And also the really heavy financial burden in our society, reaching $290 billion just in 2019, just this year. So altogether, what I wanted to just make sure I bring across to you is that this is not the disease of one individual that has Alzheimer's disease. This is something that affects families, and it's something that affects the society as a whole. There is no health system in the world, in any country, private or public, that will be able to cope with something like what's expected from now on. So this really is something important, and we really need to find a cure, something that prevents the disease, or something that even by delaying the onset of disease can have a significant impact on these numbers. And when we think about Alzheimer's disease, the most common early symptom for AD, for Alzheimer's disease, is the difficulty in remembering information that has been recently learned. And this is because the disease starts by affecting the areas of the brain that are involved in learning. And as the disease progresses, these other areas start to be affected, and people start becoming more and more confused. They start having disorientation. They are confused about people, about places, about time, and they start showing mood, behavior changes, and at the later stages, they start having difficulties in speaking and in walking. So what we have really is a relentless progressive disease. It's definitely not a part of normal aging, and we have no cure for this disease. And when we look at brains of people with Alzheimer's disease, what we see is this. Compared with normal brains, with healthy brains, you can easily see the lower volume, the shrinkage that it's noticeable in these brains. And this is due to neuronal death. So the cells in the brain die, and we have a smaller volume in our brain. And we have around 100 billion of these neurons or brain cells. And each of these cells are like tiny factories. They get supplies, they construct things within themselves, equipment, they get rid of trash. So all of this needs to be running smoothly for everything to work in our brains. And when something is not right, when some of these parts are not working correctly, we start having problems. And we think that the most likely suspects in causing this damaging and these problems are these abnormal structures that we call plaques and tangles. And these plaques are formed of a fragment of a protein, of the APP protein, which is called beta amyloid, that actually is very sticky and just deposits outside the nerve cells. And also we have inside the nerve cells another abnormal structure called tangles, and these are formed by twisted fibers of this tau protein. So when we look at the brains of people with Alzheimer's disease using a microscope, this is what we see. And this is really important for the diagnosis. But when I'm talking about Alzheimer's disease, usually the main question, or the more frequent question that I get, is what's the difference between Alzheimer's and dementia? And it's very easy to answer this. Alzheimer's is the most common form of dementia, and it's very easy to visualize it. If we think of dementia as an umbrella, that actually it's an umbrella term that it's used to include a series of different conditions. All of them have some changes in memory. The most frequent in the population of these conditions is Alzheimer's disease, but there are others, like frontotemporal dementia, Lewy body dementia, and you'll hear more about this form of dementia further ahead from Jose. And when I have a question, usually what I do is I just Google it. And if we do something like that, and we Google Alzheimer's disease, we start getting questions like, does Alzheimer's cause death? And yes, it does. Does Alzheimer have a cure? No, it does not. But we also see many questions that relate with Alzheimer's running in families. So it is a big worry for families to know how genetics works in Alzheimer's and dementia. So I'm going to talk a little bit more about this. And in general terms, what we have for the genetics of Alzheimer's is this here. We use 65 years as a cutoff between what we call late and early Alzheimer's disease. So people that develop the disease after 65 years of age, we say it has late onset Alzheimer's. If before 65 years of age, we say it's early onset Alzheimer's disease. The majority of cases of Alzheimer's disease in the population are late onset, and they are sporadic. There's a combination of genetic and environmental risk factors that combined lead to this complex inheritance of late onset Alzheimer's disease. But then we also have some early onset cases. Some are also due to this combination of genetic and environmental risk factors, but we have a minority of cases that are inherited and are caused by mutations in genes. And these cases usually have a much earlier onset that can happen as early as they're 30 years of age. And I'm going to start by talking about this part here because we can learn a lot about this. We can learn a lot when understanding the disease in these families, these early onset families. And the genes that we currently know as being involved in these forms of disease are called presenilin 1, or PSEN1, presenilin 2, and amyloid precursor protein, or APP. And I talked a little bit about the fragment of this APP when I talked about plaques before. Part of this protein is actually what gets sticky and just deposits in the brains. The first story that I'm going to tell you about, it's a good example of how genetics can be important and how studying these families can be important. And it's a story coming from Colombia. And if you're a fan of the TV series Narcos, you'll know Pablo Escobar, and you'll know that there is a lot of drug trafficking going on in Medellin and in this area of Colombia. And in addition to all the guerrillas and all that is going on in that area, they also have to deal with a very high frequency of Alzheimer's disease, of familial Alzheimer's disease. And this actually is a very unusual setting. When we have families, if you remember, I said this is the most uncommon form of the disease. But here we have 25 extended families with the same mutation, being caused by the same variation in the genes. We usually have maybe 1, 2 families across the world that share the same mutation. This is really atypical. And this is really catastrophic for the families because you can imagine having a parent and children as early as 10 years old, they start being forced to be the primary caregivers for these families. They have to take care of their parents, they have to take care of their grandparents, uncles, aunts. So there's really a very strong effect, a very nasty impact on these families. And they are now starting at the university, they are starting programs of education, trying to teach these kids how to care for their parents, trying to care for their grandparents, and how to understand the risk that they have for this disease too. So this is really bad for the families, but it can be really powerful for research because we have a very homogeneous population that we know came from as early as the 1600s when Colombia was found by a Spanish conquistador that somehow brought this mutation into and got propagated through generations. So we have a very homogeneous genetic setting here, and we know which of these individuals are going to develop Alzheimer's and which ones are not. So we can test drugs that are being developed in this setting. And this is important because many people believe that the drugs that are currently in clinical trials and that have been failing clinical trials for Alzheimer's disease, many people believe that this is happening because they are being given to the patients too late in the progression of their disease. And if you are able to give these drugs very early in the progression of disease, and this usually when people show symptoms and signs of the disease, the disease process started almost 20 years before that. So microscopically, we can see changes in the brains 20 years before the disease actually manifests itself. So if we are able to get into this space of time here, we have better chances of getting a drug that actually works for the disease. So that's what's happening here. These families are enrolled in a clinical trial that is testing these drugs and hopefully will give us some positive results soon. It's very important also to know these mutations, all these genes and how they work, because this gives us a better understanding of the biology of disease. And this is just an example. So this is called the amyloid cascade hypothesis, and it's really an hypothesis that has been underlying the research for Alzheimer's disease in the past 3 decades. This is a very simple way of putting it, but by knowing that the mutations in those 3 genes are the ones that are causing disease in the families, we know that this leads to an abnormal processing of the protein APP, leading to that sticky fragments of the protein that stick into the outside of the brain cells, leading to those tangles, neuronal death, and eventually dementia. So researchers use this to guide their research. It is also important to know how these genes work, because they can be targets for drug development. And this is another story that comes from Iceland. Until this study was published, we always thought that mutations in APP, in amyloid precursor protein gene, cause disease. But in this case, and again, because Iceland has a very homogeneous population, researchers were able to identify a variant in the same gene, this alanine to threonine change at position 673, that actually is protective against Alzheimer's disease. So this is a great finding, because if we can mimic the same effect that this variant has in the protein by making a new drug that is similar to this effect, then we are in a very good place. So I said that I was going to start with the familial early onset cases. What about the most common form, the sporadic form of late onset Alzheimer's disease? Does that also have some genetic influence? And it does, but the way we study it is a little bit different. So here we don't have causes of disease, we have association and risk factors. And risk factors just are things that increase the likelihood of someone getting a disease. They can relate to something that someone does, like smoking, or they can relate to something that it's unchangeable, like aging or genetics. And aging actually is the most significant risk factor for Alzheimer's disease. So this is a bit of a busy slide, but I wanted to show it to you because it really translates how the risk factors for Alzheimer's disease accumulate along the time and across the life of an individual, and also because it has risk factors on one side, but also protective factors, and the result comes from a balance between both these factors. What we are most interested in are these genetic factors here, and ApoE, the apolipoprotein E gene, is the most significant genetic risk factor for Alzheimer's disease. It was identified together in the 90s with those other genes that I mentioned before, but it has a different effect on the disease. We have 3 flavors of this gene called E2, E3, and E4. If we carry the E2 flavor, we are protected for the disease. If we carry the E4 flavor, we have an increased risk of disease. But neither carrying this E4 doesn't mean that we will develop the disease, and it doesn't mean that we need the E4 to develop the disease. So it's neither necessary or we don't need it to develop the disease. And what we do here is study associations. Again, we are not looking for causes of disease. We are just looking for events that occur more frequently than what would be expected by chance. And if you remember from José's slide, he said that before we were able to study 1, 2 genes or 1 or 2 variants in maybe 100 individuals. That has changed a lot, and we are now able to study a lot of variants across the genome in thousands and thousands of individuals at the same time. And the type of results that we get is something like this. And in the x-axis, we have all chromosomes, chromosome 1 to 21. Each individual dot here represents 1 variant in the genome. And here in the y-axis, we have the significance of association with the disease. So if we have a very high peak like this, we know that ApoE is significantly associated with disease. And This is called the Manhattan plot because it sometimes resembles the skyline of Manhattan. And if we think about the evolution of New York City skyline in 1920, you would see something like this, just a couple of buildings there that pop up to your attention. As people move into the city, more work is done, we start getting other tall buildings, other skyscrapers added to this skyline, and in 2020 we have something like this expected. So we have tall buildings that are being built at the moment. Some are projected, some are already there. And this is what happens with our genome-wide association studies and with the identification of these risk factors. So before we only had ApoE, but as we are able to add more samples, add more cases and more controls, and do another study, we start identifying other peaks that are also associated with disease. And today, after adding close to 500,000 samples to these studies, we know we were able to identify around 30 regions of the genome that are associated with Alzheimer's disease. Each one of these regions has a very small contribution to the risk of the disease. So why is it so important to identify this? So one of the reasons is because they point us to biological pathways. And biological pathways are just, a series of interactions among molecules in a cell that lead to some outcome. It can lead to turning genes on and off. It can lead to making the cells move, for example. And when we, as researchers, represent a biological pathway, we usually show something like this. A piece of a cell, a small part of a cell, or an entire cell where a series of things are happening, and what's represented here is the processing of APP that I mentioned before, until it gets to amyloid beta that will get deposited in the brains of patients with AD. But in reality, what happens is much more complicated. And this is actually a very small piece of how complicated it gets. So we need a way to focus our studies in the pathways that are really important. And GWAS helps us doing that. So it has confirmed that metabolism for APP is important. Tau is also important. But we also have pathways as lipid and cholesterol metabolism and immunity and inflammation coming up as important factors. And we have contributed to most of these international large studies. We have also taken a bit of a different approach, and we have started a collaboration a few years ago with a group in the University of Istanbul, and we wanted to do this to study recessive forms of dementia. We wanted to see if there were any genes that would cause dementia and Alzheimer's disease in a recessive form. And if you remember from Jose's slide before, we can have families having the disease being transmitted in a recessive or in a dominant way. Most vast majority of cases in Alzheimer's are dominant. So we wanted to test if there was also any recessive genes in there. And we studied this population because it has a very high frequency of consanguinity. So these genes are much easier to identify under these conditions. And we characterized dementia in this population by identifying how many mutations there were in presenilin 1, presenilin 2, APP, so the genes that were known already. But we also looked at all the genes at the same time in this group of individuals, and we identified 3 that had mutations in this gene called TREM2. And one of these mutations was a novel mutation, but 2 of those had been previously associated with a disease called Nasu-Hakola disease. And this is a very rare form of disease. I've never heard of this before getting these results. It's a recessive disease. You need to have both alleles, so the DNA from the father and from the mother, both need to be changed for the disease to happen. And patients usually start complaining about pain in their wrists, in their hands, in their ankles, and in their feet. And this is caused by bone cysts and bone that then, as the disease progresses, lead to bone fractures. And the patients start developing behavior changes. They die in their late 40s, early 50 years with dementia. What was really interesting about these 3 cases in the Turkish population was that none of them had these bone phenotypes. So they had an atypical form of frontotemporal dementia, one of those forms of dementia that I mentioned under the umbrella at the beginning. And so we thought, well, maybe the gene is involved in other forms of more typical FTD. So we looked at cases of typical FTD cases, and we didn't see anything. But when we moved to Alzheimer's disease, we started seeing the same types of changes in patients, but this time with only one allele being affected. So either the mother or the father had passed that change to these Alzheimer's disease cases. And we identified one variant in particular called R47H that was significantly more frequent in cases than in controls. And this is what we call a risk factor for the disease. So we identified the new risk factor for Alzheimer's disease. At the time, we were not we didn't completely understand how important this was, but this was the most significant after ApoE. And the number of studies that have been happening after this have been really incredible, with studying biomarkers, getting new research lines into industry, trying to study the types of cells where this gene is expressed. And it really, so we earlier this year went to an international meeting in Portugal, and they had this big wall of landmarks in Alzheimer's disease, and TREM2 was part of the wall, so we couldn't just help it but taking a selfie with the wall. So this was really a replicated and true finding that we are very proud of. So with this, I'm going to pass on to Jose so he can talk about Parkinson's and DLB, and you'll see that many of the features and many of the things that I have been talking about for Alzheimer's are very similar in these other diseases. Thank you.
Thank you very much. So, switching gears a little bit and talking about Parkinson's disease. Most of you will know Parkinson's disease is the second most common neurodegenerative disease following Alzheimer's disease. There's an estimated 10 million people in the world that have Parkinson's disease. It's a very common disease. It's also important to note that the vast majority, much like Alzheimer's disease, the vast majority of cases are sporadic. So there isn't a single cause for this form of Parkinson's disease. They're sporadic. They tend to occur after a person is 65 years of age, and there isn't a single genetic cause for this form of Parkinson's disease. The other 10% tend to have a genetic mutation underlying the disease. So in terms of quick facts for Parkinson's disease, I think you'll know most of this. People living with Parkinson's disease start by showing tremor. They have slowed movement. Their muscles are rigid. This leads to an impaired posture and balance. And of course, there's no cure for Parkinson's disease, like there isn't a cure for Alzheimer's disease. When we look at the brains of people that lived with Parkinson's disease, what we see are these protein deposits inside brain cells. And these protein deposits contain this protein called alpha-synuclein. And alpha-synuclein is a protein that's encoded by the alpha-synuclein gene. And this makes up this structure called a Lewy body. And this is what really characterizes Parkinson's disease when you look at the tissue that's affected by disease. So genetic comes into Parkinson's disease early on, but I will say that when we started to read about Parkinson's disease many years ago, actually it was thought that genetics would not play a role at all. And if you had any history of this disease in your family, you could not have Parkinson's disease. This was an exclusion criteria for a diagnosis of Parkinson's disease. And now we know that that's not correct. We know that genetics plays a very important role. And we've known that since about 1997, 1998. But in about 2004, we already knew that there were a handful of genes where changes, mutations that are very rare in the population, so they're on this end of the spectrum here, they're very rare, but when they occur, they have a very high effect on disease, and they tend to cause disease when they happen. We've known this since about 2004. And as the years went on, and technologies were developed, we started to be able to study other genes that are more common in the population, so they're towards this end of the spectrum here, but have a lower effect on disease. So they're more common, but when they occur, they do not cause the disease. They only increase risk for the development of disease. And so over the last few years, we've really been able to build up this picture of the genetic architecture of Parkinson's disease. And today, we're somewhere close to this. This is actually incomplete, and I'll show you in a couple of slides why this is incomplete. So there's a lot more genes here that we know have changes that are common, and they only increase a person's risk. They don't cause Parkinson's disease. There's a larger number of genes here where rare mutations tend to cause disease. When they occur, you tend to get Parkinson's disease. But also importantly, we found a couple of genes here in the middle where changes are not very common in the population, but they're not very rare either. And they have also an intermediate effect in terms of risk. So they have a higher effect than these genes here, but a lower effect than these genes here. And one of these is GBA. And just remember that name. I'll come back to GBA in a couple of slides. So the genes we find here, they're found by association studies, the type of studies that Rita mentioned, and I'll just bring this up to remind you of what these studies are. So what we do is look at genetic changes in the DNA. So let's imagine this is a little bit of DNA, and we want to see if this change here, if this T is involved in disease. And so what we do is take a group of people that are living with Parkinson's disease and a group of people without Parkinson's disease, and we see if this change is overrepresented in Parkinson's disease when compared to people without. And if it is overrepresented, then we say that it is associated with disease. If it occurs more in people with disease than in people without, we say it's associated with disease. We're not able to say how it is associated, what it does, but we're able to say that it is associated. And so that's the type of study that we do to find risk factors for disease. And I say this because the most recent genome-wide association study, which is how these studies are called, in Parkinson's disease was very recently published. We were part of this as an international consortium. And in this study, we had almost 38,000 people with Parkinson's disease, and we looked at their genetics, and we compared their genetics with about 1.4 million people without the disease. So a really large study that would only be possible through international collaboration. And in this study, what we found, and this is the Manhattan plot that Rita showed before with the New York skyline, what we found was that there are 90 genes that are involved in changing risk for Parkinson's disease. So when I said that the orange figure before was rather incomplete, it's because there's so many genes now that if we were to put all of their names there, we wouldn't have space. So there's so many genes that we now know are involved in modulating a person's risk to develop Parkinson's disease. The amount that they increase a person's risk individually is tiny. It's very, very small. But altogether, we can measure the effect that they have on disease. But also, very importantly, each one of these genes that we find has the potential to give us a new therapeutic target. If we know that gene is involved in disease, and we know what the protein that gene encodes does in the cell, this gives us an avenue of research into developing novel therapeutic approaches. And I'll give you one example, a great example of this, which is GBA. The gene I just a few slides ago told you to remember about the name. GBA is an incredibly interesting gene. We've known that GBA mutations, when they're homozygous, cause Gaucher disease for a number of years. We've known about this for a number of years. So Gaucher disease is a recessive disease. It's a disease that starts early in life. It's not like Parkinson's disease. This is an very early onset disease. And what happens is that in the cells, in all of our cells, we have these components called lysosomes. And lysosomes, their job is to clean up waste from the cell. So what they do is to clean up debris from the cell. They do this because they have a number of proteins inside that work towards this end. One of those proteins is the protein encoded by the GBA gene called glucocerebrosidase. And when people have two mutations in this gene, what happens is that the lysosome doesn't work. And so stuff just accumulates in the lysosome. And this leads to cell damage. So the cell doesn't work as well as it should because there's all of this waste inside of it. So what was found, and this was found by Alan Sidransky, and I'll just do a brief side note here. Alan Sidransky was just awarded this year's J. Van Andel Award for Outstanding Research in Parkinson's Disease, and she'll be joining us for our Grand Challenges meeting in August here in this auditorium to tell us about her research on GBA and the most recent research that she's doing. And so I just wanted to briefly mention that if you're interested in this type of work. So what Ellen did was to identify that when you have not 2 mutations in the GBA gene, but a single one, you don't get Gaucher disease, but actually you're at higher risk for Parkinson's disease. This was puzzling at the time because Gaucher disease and Parkinson's disease are very different diseases. One is a neurodegenerative disease, the other is a disease that occurs very early in life. So they had nothing to do with each other, apparently. But actually, the mechanism that's involved in both of these diseases, or at least in some cases of Parkinson's disease, it's the same or very similar to the mechanism that occurs in Gaucher disease. And this is important because we have a number of therapies for Gaucher disease patients. And this suggests that perhaps we can test some of those therapies in Parkinson's disease as well. And that's actually what's occurring. And there's a number of those therapeutic agents going into clinical trials now. So this is what I wanted to tell you about Parkinson's disease. I'll now switch gears a little bit and tell you about Lewy body dementia. So Lewy body dementia is also a form of a neurodegenerative disease, and it's the second most common type of dementia after Alzheimer's disease. And I'll bet that the majority of people here have not really heard of Lewy body dementia in the past. I may be wrong, but I tend to gamble, and I tend to be right when I say this. It's a very common form of dementia. There's well over 1 million people in the US that have Lewy body dementia, but at the same time, it's an unrecognized disease. People don't know about it. Research hasn't really occurred that much in Lewy body dementia. And one of the reasons for that, it's not the only one, but one of the reasons for that is that Lewy body dementia is very often misdiagnosed. And it's misdiagnosed because a patient with Lewy body dementia has symptoms that resemble very much those of patients with Parkinson's or Alzheimer's disease. And so when they go to their neurologist, because Parkinson's and Alzheimer's are more common, they tend to get the diagnosis of one of those diseases when it's actually not correct. So we've been getting much better at identifying these patients, but there's a substantial proportion of people that have Lewy body dementia that are diagnosed as something else. So the misdiagnosis rate is very high. Much like dementia, Lewy body dementia is an umbrella term. It encompasses 2 diseases, dementia with Lewy bodies and Parkinson's disease dementia. They're very similar, these 2 diseases, DLB and PDD, they're very similar. But we as scientists like to categorize things, and so we came up with a way to separate these 2 diseases, and we do it by the onset of dementia in terms of time of disease. If dementia occurs before Parkinson's disease symptoms, we call it dementia with Lewy bodies. If dementia occurs after the onset of the movement disorder, we call it Parkinson's disease dementia. We don't know if the disease itself is different, but there are differences in the temporal onset of dementia. And so we categorize those 2 forms of disease like that. When you look at the symptoms that are associated with Lewy body dementia, you can see very clearly that they match up very well with what we've seen for Alzheimer's disease from Rita and from Parkinson's disease in the last few slides. So it's easy to see how a misdiagnosis can occur in these forms of disease. And even when we look at the brains of people with Lewy body dementia, what we see are Lewy bodies. And Lewy bodies, as I've shown you before, are what tells us that a disease is Parkinson's disease. And so there's a lot of similarities between these diseases. Now, if you have heard about Lewy body dementia or dementia with Lewy bodies, which I'll focus a little bit more from now on until the end of the presentation, if you've heard about it, it's likely that you've heard about it because of Robin Williams, who, when he died, it was found that he had Lewy body dementia after he died, or because of the news that Ted Turner last year also revealed that he has Lewy body dementia. This is how most people have come across this term, Lewy body dementia or dementia with Lewy bodies. And this is also true for, in a way, for scientists. So this is a plot showing the number of scientific publications for each one of these diseases over the years. And so you can see that Alzheimer's disease is this orange color here, and there's, just last year, there were about 10,000 scientific publications with new data for Alzheimer's disease. For Parkinson's disease, it's well over 5,000 just last year. This isn't cumulative, this is just by year. And so a substantial number of publications for both of these diseases. But when you look at dementia with Lewy bodies, last year there were less than 500 papers. So it's not only that people don't really recognize this disease and how important it is, but scientists over the year actually haven't recognized it either. And so there's a lot to be done in Lewy body dementia and dementia with Lewy bodies, specifically because it's such an important and common disease. So in 2014, we had a look at the genetics of dementia with Lewy bodies. This was the first time that a large-scale study was done, a large-scale genetic study was done in dementia with Lewy bodies. And what we found was that one of the genes that was associated with dementia with Lewy bodies was this SNCA, and this is the gene that encodes alpha-synuclein. This is the protein that's in Lewy bodies. And so we're very excited about this because there was an obvious link. We found a gene associated with the disease where the obvious problem in the brains of people with this disease is this protein. So we're very excited about this. We're also excited because the same gene is involved in Parkinson's disease, and so it all made sense. Lewy bodies exist in both diseases, and we find the gene associated in both diseases. So that was very interesting. But then we went in and looked at the gene with a little bit more detail. And what we're looking at here is just a very zoomed-in view of the Manhattan plot that Rita showed you before. So we have the position in the chromosome here, and we have the association strength in the y-axis here. And what we're looking at, each one of these dots are markers, are positions in the DNA. And the higher they are, the more associated they are with disease. And if they're red, they're for dementia with Lewy bodies. If they're blue, they're for Parkinson's disease. And what was obvious to us when we looked at this was that the more associated markers with dementia with Lewy bodies were different than the more associated markers with Parkinson's disease. So despite the fact that the same gene is involved in both diseases, it's different parts of the gene that's involved. And this we don't know what this means yet. We're working very hard to try and understand this, but it gives us a difference. There are different diseases, the same gene is involved, but the way in which it is involved is different. And so this, we think, is going to be very important to understand for us to get a better grasp of what happens in dementia with Lewy bodies and Parkinson's disease. We did a number of other studies looking at genetics. The most important thing here is that we've shown that the genetic component of dementia with Lewy bodies is as important as the genetic component of Parkinson's disease, which we know is incredibly relevant. So we've shown a few years ago that genetics plays an important role in dementia with Lewy bodies. And just last year, we've published the largest association study in dementia with Lewy bodies. We had about 2,000 cases and 5,000 controls, and we found 5 genes associated with this disease. It's not the 90 that we have for Parkinson's disease, so we're still in 1982 in terms of the Manhattan skyline, but we're getting there. We're certainly getting there, and this is very exciting data for us. So there is a clear genetic component to the LB. But when we take all of the genetics we know occurs in Alzheimer's disease, and all of the genetics we know occurs in Parkinson's disease, and we see if we can distinguish a DLB person from a control, we see that we cannot. And so this tells us that DLB, the genetics of DLB, tell us that it's very likely that this is a separate disease from Parkinson's and Alzheimer's disease. So there are all of these similarities in symptoms, in the pathology, there's all of these similarities. The genetics seems to tell us that they're actually different diseases, and this is very, very important. So in terms of way forward, we know how to do the next things that need to be done in terms of genetic analysis. We can identify common changes that increase risk for a disease. We can identify rare changes that cause these forms of disease. Some of these will be very rare. They'll occur in a single family in the entire world, but we can identify them. There are things that we need to be better at. So we say that a gene is associated with disease, but we still can't often say how it is associated with disease. So understanding those hits from an association study is something we need to work towards. Also, genes that modify disease. Not all Parkinson's patients, for example, present the same way. Some have slightly different ways of presenting their disease. And we think that genetics plays a role in that, in modifying the disease. And so we're working hard at identifying those genetic underpinnings. The same thing for progression. Some people progress through disease quicker, some people slower. If we can identify what genetics drives the slow progressors, maybe we can come up with a therapy that does the same thing, and that would be very important. And also, as Rita shown, protective changes, mutations that occur that protect you against these diseases. These will also be very powerful in helping us find therapies. And of course, as we get a better understanding of the mechanisms that occur in these diseases, we'll have a better chance at identifying therapeutic interventions, because that's what we're here to do. I mean, we as scientists like to pose questions, so it's interesting to know how a disease occurs, because it's a question we have, how does this occur? But actually what we're here to do is not to understand the disease, we're here to come up with therapies. That's what we're doing. We want to have a therapy that slows or stops these diseases. And so identifying genes gives us targets that we can try and hit with a therapy. And the more genes we find, the more targets we have. And over the last few years, we've been very successful at identifying more targets, and this will improve as time goes on. And I would say that these are very exciting times. When you're studying these diseases, it really feels like we're at the brink of making a very significant discovery. And so I think I'll end it there. I'll just leave the slide. If you're interested in these types of things, the next round of our public lecture series is going to be on September 25th. We'll have Matt and Stephen talk to us about translational medicine. And if you have your calendars with you, I'd suggest you pencil in this date.
This technically concludes our lecture. We want to thank you for joining us today. It is our privilege to bring to you, as you can see, we often say we're an institute with walls and without walls. You can see the important role that global collaborations play, and we're thankful for the Van Andel family who established both the institute as well as some of our funding. But we're dependent upon folks like yourself and many others that support the research that these fine faculty do every day to attack debilitating diseases.